Longitudinal biomedical data holds immense potential for early disease detection, metabolic disorder monitoring, and even identifying illicit substance use in sports. However, detecting subtle anomalies in this data is challenging. Current methods often fail to integrate temporal dynamics, biochemical knowledge, and explainable reasoning into a unified system. This is where CASPIAN comes in. CASPIAN is a cutting-edge framework designed to address these gaps by combining structural temporal analysis, metabolic pathway reasoning, and population-based analysis. We'll explore how CASPIAN works and demonstrate its application in detecting doping in sports. In high-stakes scenarios like anti-doping analysis, missing anomalies can have serious consequences. For example, undetected doping cases undermine athletic fairness, while undiagnosed diseases risk patient harm. Traditional methods often treat longitudinal data as static snapshots, ignoring temporal dependencies and biochemical context. This leads to missed anomalies or biologically implausible results. CASPIAN addresses these challenges by integrating 4 specialized modules: SACNN for structural temporal anomaly detection, GRAMP for metabolic pathway validation, MPP for domain-specific reasoning, and DAP for population-level analysis. Let's dive into each of these modules. The first module, SACNN, uses a self-attention-based convolutional neural network to analyze longitudinal data. It captures both structural dependencies between biomarkers and temporal dynamics over time. It processes irregularly sampled data by generating fixed-length subsequences, allowing it to handle variable sequence lengths common in clinical monitoring. For example, let us analyze the longitudinal steroid profile of an athlete. We will use a dummy profile for this demonstration. Firstly, the user can upload the data into the software. Next, the user can define the specificity to operate the model. The SACNN model will run and compare each sample with other samples in the longitudinal profile. Here, you can see a set of subsequences with the respective anomaly predictions. SACNN can detect a sudden spike in testosterone levels that deviates from an athlete's historical trends. This temporal anomaly is flagged for further analysis. Next, the GRAMP module models biochemical interactions as a directed graph, where nodes represent biomarkers and edges encode enzymatic reactions. GRAMP validates anomalies flagged by SACNN against metabolic plausibility. For instance, if SACNN detects a spike in testosterone, GRAMP checks for structural inconsistencies in its precursor metabolites. If the observed metabolite levels deviate from expected enzymatic conversions, GRAMP identifies the anomaly as biochemically implausible, signaling potential fraudulent activity. Next, we have the MPP module, which provides contextual reasoning for anomalies flagged by SACNN and GRAMP. It integrates biochemical domain knowledge to generate human-readable explanations. For example, MPP might explain that a testosterone spike coincides with suppressed 5-alpha reductase activity, disrupting the expected conversion of testosterone to 5-alpha androstenediol. This pattern aligns with exogenous testosterone use, providing actionable insights for anti-doping agencies. Finally, the DAP module provides unsupervised anomaly detection by analyzing an athlete's longitudinal profile against population-wide trends. It transforms multi-biomarker profiles into a simplified, variance-preserving space and flags outliers exceeding population-derived thresholds. For example, DAP transforms the athlete's steroid profile into a 3D principal component space, where it calculates the distance of the anomalous sample from the centroid. In this case, The distance of 25.8 exceeds the individual threshold of 6.2, confirming the anomaly. By integrating SACNN, GRAMP, MPP, and DAP, CASPIAN provides a robust, interpretable solution for longitudinal biomedical anomaly detection. Its application in anti-doping analytics demonstrates its ability to detect sophisticated fraud activities like sample swapping while reducing false positives compared to traditional methods. Future work will expand CASPIAN's scope to neurodegenerative disease monitoring and refine its adaptability for diverse biomarker profiles. CASPIAN represents a significant step forward in precision healthcare and sports integrity. Thank you for watching this demonstration of the CASPIAN framework. Together, we can advance personalized biomedical analytics and ensure fair play in sports.